start the webinar in case we have guests. Marco, it's good to see you. We are now live on the internet, on YouTube, on LinkedIn, and on several recording stations to make sure that we don't lose any information of this valuable podcast. It's good to see you today. Yesterday, when I prepared for the episode, I saw a post on LinkedIn from Lex Friedman. He's a podcaster from the United States. And he wrote, maybe I can share my screen and can show you the post. He congratulated DeepMind for developing Alpha Tensor. And he said, these are the early steps in AI, inventing new ideas in math and physics, worthy of a Nobel Prize and Fields Medal. We live in exciting times. And this morning, I pulled up the Nature publication, the article he referred to. DeepMind's AI invents faster algorithms to solve tough maths puzzles. Did you hear about that? I mean, you referenced DeepMind in your article on LinkedIn.
Yeah, so there's the company DeepMind, but I mentioned Alphafold. Alphafold is a deep learning application to predict the protein structure just based on the amino sequence. I need to say that I haven't read the article, so but it looks for me that this is an other area where is DeepMind active. But I'm only aware of Alphafold now. Sorry.
Yeah, but it's pretty exciting, the development of Alphafold as well as the latest article, and I'm curious to hear more about what can artificial intelligence do in drug development and drug discovery. Let's start with the first question. Can you please give me a little bit overview of where you're coming from, what your profession is, and what your company is doing?
Yeah, so my name is Marco. I'm originally a biochemist. So I studied biochemistry here in Germany, in Tübingen and in Berlin. And then I went on to conduct a PhD in fragment-based drug discovery. And this was then followed by a postdoc at the University of Cambridge, mostly founded by the Bill and Melinda Gates Foundation. So I moved more and more into the field of drug development, of drug discovery in my academic career. And when I worked in Cambridge, I need to say that I worked with brilliant guys. So Chris Abel, the late Chris Abel, died 2 years ago, unfortunately, and Tom Landell. And these guys were the SDEX founders. So they founded a company called SDEX in the fragment-based drug discovery field, I think 1999. And then they sold this, I think in 2013 to Zuka. And what I learned from these two guys, and especially with the setting with the Bill and Melinda Gates Foundation, is that the big problem is not to find the ligand for a drug discovery or a drug development project. It's more problematic to predict the clinical efficacy of the ligand. So this is actually the black box. So in the beginning, when you think about the disease, how you want to target the disease, and all the models you use, so you read a lot of literature, you look at in vitro experiments and so on. But these are all small experiments, and the big final experiments, when you go into human and you really want to see whether there's an effect or not, there's actually no prediction available. So this is actually a black box. And in 2013, there was then the first publication by the former head of R&D of US Merck, so Edward Skolnick, and he proposed the concept of genetic support or genetic evidence. So he said, when the drug target, so normally you have a ligand, and the ligand binds to a drug target, to a protein, mostly a protein, and modulates the function of the protein. And when the protein is somehow connected in a genome-wide association study with the disease, then he told this drug target linkage, the genetic evidence. And if you have it, he postulated, then it's more likely that this drug target is causal for the disease. So modulation of this would have an effect in the clinical trial. And this was then proved in 2015 in a big retrospective study by people from GlaxoSmithKline. So they looked at all successful and unsuccessful drugs in the last 30, 40 years, and they really could show that there's evidence. Then when you have genetic evidence, when you have genetic support, then it's more likely, I think it's 2 times more likely that there is efficacy, and efficacy is then shown in the first, also in a clinical trial with patients. But the point already is that was just what I call explanatory statistics. So you look at data and you explain the data. Here at Biotx.ai, the idea was then when I met my co-founder Jörn, he's a computer linguist. So I'm the biochemist, he's the computer linguist. Was the idea to use predictive statistics, what is now called machine learning or artificial intelligence. So just another kind of thinking. So we try to find now the patterns in the genetic evidence, to use this for prediction whether a drug and a drug target combination will be successful in a clinical trial for a specific disease or not. And yeah, that was the idea in 2015, 2016. We had then here a so-called startup grant at the University of Potsdam. After this, we then joined, and that was, I really have to say, we joined Startup Bootcamp Digital Health Berlin, so an accelerator program here. And then we also founded then Biotx.ai, our company. And yeah, now we're working more than 4 years on this. And
congratulations.
Or 5 years, sorry, 5 years.
So you are over the worst phase of a startup. So the first 3 years are crucial, and then comes the big success.
Yeah, actually. So, and we turned also from the business model from a service provider into now our own products. So we have now 3 products in clinical development and 1 clinical trial running. Yeah.
How much money did you raise so far, out of curiosity, if it's not confidential?
It's not confidential. So it's actually not so much as 3 million. And the rest is then was revenue based. It's
very efficient. I had 2 recent podcasts where I was more focusing on entrepreneurship, on investing, and they're also doing research and development in artificial intelligence. Let's stay in this podcast episode on the artificial intelligence side, and let's start with some basic terms and with some basic definitions. I know from the last year that many retail investors, business angels, got curious with the successes of CRISPR and Intellia to invest in life science projects. But it's not the most easiest field. I mean, let's start with the first term that you mentioned, drug development and drug discovery. When I entered the life science industry in 2006 and I told people that I'm developing drugs, they looked at me and said, "What? Are you doing something illegal?" And to avoid in the times of cancel culture that this podcast episode is taken down, let's start with defining drug development and drug discovery in the context of this episode. What is your definition of drug discovery and drug development?
So first of all, so to make it clear, so this is not illegal. I know that these 2 words, because when you're German speaking is, then there's often a misunderstanding. So here I would like to focus, we are working on medications. So we are working on different kinds of chemical or biological entities that can modulate some stuff in your body in order to treat or cure a disease. Then the next, what you asked, drug discovery and drug development. So normally discovery means that you're really in the early phase of your development. Discovery means then that you still think about the disease, what are the mechanisms of the disease, how you can treat the disease. And then you start developing ligands, try to test the ligands in vitro models, so in disease models very easily. And that's why there's still this discovery thing, because it's like the land is not completely, it's still unknown and you are the first entering it. Development is a little bit more when you have already proven that your ligand works and you want to go into the normal way how to prove it. And you can say there are, I call it now 5 phases. The first is so-called preclinical phase, where you check your ligand in vitro models that it's safe. The next one is the so-called phase 1 clinical trial. And here it's still looking at safety. So you tested them in humans, and you're very interested that these humans, they do not have any severe side effects. So you tested actually in healthy humans to check the safety. And then the clinical phase 2 becomes, from a development standpoint, much more interesting, because this is the first time when you have this so-called dosage finding. So you want to think about what is we call it a therapeutic window. So what kind of dosage you have to administer, which shows an effect, but is not toxic to the person. And here in this, especially in the later phase 2 clinical trial, because this phase 2b is the first time when you also look for efficacy, for the efficacy signal. That's why this is the biggest problem. So the normal success rate in a phase 2b clinical trial is 29%. So it's extremely low. It's extremely low. And then there's a clinical phase 3, and this is mostly the big cohort where you have to show from a statistical point of view that there's efficacy. So you need a lot of patients and healthy controls. It should be randomized, placebo-controlled, prospective to really show that there's an effect. And nowadays we have also what we call phase 4. And phase 4 is after the approval that your medication is still under observation by you as the developer and also by the authorities to look whether there are any side effects that are now in the usual way, how you in the clinic, in the daily routine, is there probably some difficulties you haven't seen in the testing. So these are the five phases, and the 5 phases you normally say this is the drug development phase, and everything before is the so-called drug discovery phase.
That's an excellent definition. Thank you very much. Let's also define a second word that you use quite frequently in this episode and that people interested in investing in drug discovery and drug development projects you might also hear in the future. It's ligand. In the context of this episode, what is a ligand in drug discovery?
Yeah, so what is a ligand? I would like to say it in a very simple words. It's actually the tool, and it's from a business perspective, your asset. So a ligand can be a chemical structure, or it can be also a biological entity like an antibody or a peptide. But this is actually something what you have, I call it, on what you have on your desk and what you have to take. So this is actually your tool. And the ligand, it by the definition binds to a target, to a drug target. So mostly it's a macromolecule with mostly with deep pockets where a ligand can bind into. And this, by binding, there is then a change in the function of this macromolecule of the protein, for example. And with this modulation of the function, you want to induce an effect in the body. For example, very famous are the statins for cholesterol. They bind to an enzyme which is responsible for cholesterol synthesis, and they block the active site. So no cholesterol is anymore produced in the liver, for example. So the ligand is your tool in drug development and drug discovery, and is from business perspective also your asset, because you only can predict it.
I like the business approach that you have. I really enjoy this definition. A ligand is your asset. Whenever you hear a presentation and a scientist is talking about a ligand, listen closely. He's talking about your asset. So this is the most important part of the presentation. I think this is great. When we stay in drug discovery, I think a lot of people now know drug discovery from the last 3 years. We heard all the BioNTech and Moderna stories with phase 3 studies with 40,000 patients, and also the price point of 3 to 4 billion. And when you look at the length of drug discovery and drug development process, I think the early days of mRNA technology were in the 60s, and BioNTech started somewhere in 2008, so they were working really hard for over 10 years on moving the asset forward to a stage where they can produce a new vaccine over the weekend. So this was in the news. Why is this process so expensive, so long, and so challenging?
Yeah, there's a lot of thinking about this, and I don't know whether I'm the right person about this, because I think I'm still too young, that I have not thought about so thoroughly as other people do. My personal view, and this is just my personal view, is that the way how we do drug development, drug discovery is still very what we call reductionistic. So in science, in the last 200 years, we have this mindset of reductionistic science. So what does it mean? When you have a big problem, you separate the big problem into smaller parts, into smaller problems, and then you try to solve these smaller problems. And when you have the solution of these smaller problems, you think you can put these smaller solutions together, and then you have the big solution. So this is these, and this is very, in my opinion, very, very successful, this reductionistic approach. So think about your problems, how you approach this, and it's often that you start separating the big problem into smaller ones and then put it together. The problem is with living systems, especially with the human body, is that it's more than its parts. So, and this is, in my opinion, the big problem. So we always see that we tend to have very simple models in biology, because the biology is extremely complex. Our tools are also very limited. Then it's still to say it, in my opinion. So you have your pipettes, you have your in vitro assays, and so on, and you tend to have very simple models. And then based on these simple models, you have an idea whether how the ligand, how the drug is behaving in your body. And when you then go, for example, from your cell dish to an animal model, it's always awkward. It doesn't work, because still the animal is more complicated than I expected it. And then the next step to the human body is also very, very complex. It's very, very difficult to apply this from a very simple model then to the human body, because there's in the human body, is a lot of black box. There's a lot of things we do not know, that we do not understand, but we really want to manipulate the system in a specific direction. And this is it, in my opinion. This is what makes it so difficult and very risky and so painful, because if you do not understand the machine in its whole, and you want to manipulate it, then it's extremely difficult.
Yeah, that's absolutely true. The human body is still a mystery. When I just think, I read some articles recently about longevity research and the effects of sleep, nutrition, of exercise, life habits. I think this all has an effect on the biological system, and it changes also the environment for scientists like you in different patient populations. It's absolutely clear. When I look at the success rates, I think in drug discovery and early stage drug development, we are talking about 99% failure rate before a project reaches the clinic. So 1 out of 100 makes it, and the other 99 are for publications and to increase the know-how and the knowledge. And when I look into the clinics, I think it's a little bit better, but we are still from a phase, I think, as far as I remember, from a phase 1 project, from 10 phase 1 projects, 1 reaches the market. So, and the other 9 don't.
Yeah. But you have to differentiate. So big pharma companies are actually not so good in R&D productivity, but biotech's much more better. So you see here, I don't know whether you heard about this famous word, it's called erooms law. So it's a little bit, no, you haven't heard about this?
No, it's, explain it, please.
Okay.
Tell me more.
So this is actually the complete opposite of Moore's law. Moore's law is, when you look at CPUs, they become better and better over the time. And erooms law is the point that the R&D productivity is decreasing over the time.
Really?
Yeah. So this is the overall idea of erooms law, or how they describe the R&D productivity in big pharma. But what we have seen since 2010 is that they call it breaking erooms law, that the productivity is now increasing also in pharma. And this is very interesting because we see now that there's a lot of change in decision-making in pharma and in biotech based on novel technologies. And it's also other business strategies we see that makes these projects more successful in the past.
That's amazing. I didn't look at it from this angle. I just looked at it from the organizational point of view. And my personal explanation why R&D doesn't work so well in big pharma companies is that, by definition, a company is an organization where more than one person works hard on accomplishing just one goal for a defined set of customers. Just think about Apple, for example. Apple, for most of the time in this century, was a one product company that just delivered Apple iPhones, smartphones. And then their organization is set up to just accomplish this one goal, push one smartphone to the population. And every time when something new is discovered, it means that this new thing that might be beneficial in the future is simply outside of the mission of the company. So it means doing research and doing R&D inside the company can't work by definition because it doesn't really help the current goal and the current mission. So it might help in the future, but counteracts the processes that are set up in the company. And my personal opinion always was, when you find something new, put it in a startup outside of the big organization, set up a project team, let them work on it. And when they are successful, then you can reintegrate it because then it fits into the mission.
It's a little bit the innovators to deliver a dilemma you're talking about, I guess. So that for a big company or for a big organization, a new technology is always a kind of a threat to the existing business model.
Yeah, you could call it that way. You could call it that way.
Yeah. I don't know. In my opinion, when I look at a pharma company, especially R&D facilities, in my opinion, it's somebody with their job to do to prove new technologies. And people are always complaining about pharma, but in my opinion, it's tough. And in the past, they had too much focus on these reductionistic tools, what I explained before. But you can say, so this the Human Genome Project in 2000, I guess, so mid 2000, you see more and more that they are using genomics, omics technologies more and more. And they are also going more into very specific disease areas. So they break with the general idea of the big blockbuster. So where you have 1 million patients, and when you want to deliver a drug for 1 million patients, then I call it the things you have to do. It's very tough. You have to conduct a big clinical trial. You really have to show that the endpoints work and so on. It has to be safe in a broad population. And since 15 years, I would like to say, we see that they are more going for, I call it, diseases with smaller patient groups, more targeted, where they have more omics evidence that this is working. And here you really see that this is more successful. For sure, a biotech is always faster in moving. But for example, when you see Roche with Genentech, for example, they try to set it up that they have this more startup-oriented organization with Genentech, and you have still Roche with the traditional pharma and of course sales and distribution units you need when you want to market the drug.
Genentech is a good point. I'm currently reading the book of Sebastian Malaby, where he is talking about the power law and the development of venture capital in Silicon Valley, and Genentech is a big part of the book. I mean, you mentioned it. I think I like also the term networked economy that exists since the 90s. I think trying to put everything in a big company, so from the beginning of the research and development up to marketing a product, is just a too diverse mission. You mentioned marketing and sales, bringing a product to, like the mRNA vaccines, to 1 billion or 2 billion of people in different jurisdictions is hard work. And I would rather focus a big organization on this goal to distribute the finalized product than trying to push everything in the entire value chain. So I really like the setup that we have currently. You have big pharma that are proficient in marketing and doing the final mile with the regulators, then the early stage biotechs that solve the development problems that are not really suitable for research organizations due to a different mindset, and also not yet ready for big pharma. And then you have the early ground of pure science and research, where I think the research organizations are the best part to have it. So I really like this value chain approach. We have research, you have the market experts, and in between there are biotechs and venture capital.
Yeah, but they're also a problem for the biotech company, because the value creating step, or where you make the money, is still in pharma. So there's this famous idiom, when you have invested $1 in a biotech stock and $1 in a pharma stock, then the biotech, your biotech investment is now worth $15. And when you have invested it in pharma, it's now $800 worth. So you see, and this is actually one thing at the biotech people have to think about, how they can capture the value still, because all the value is created in the last step and all the money is earned in the last step. So it's then very difficult to keep, or how you can keep up with the success in the later stage with your ideas in the early stage.
Negotiation talent in between.
Yeah,
interrupt me. Just go ahead.
No, what I mean is, you see, and then we can also apply this to the AI drug discovery development phase industry. So the startups in this area. So they all started as a service provider, as we did, for example, and then you move over the time into a clinical stage company, because you see the real value is at the end. And there was, again, 2 weeks ago, there was really nice article by the same guy, by the same person who coined the name erooms law, Jack Scannell, and he published a game theory paper about why all the biotech companies, the platform, especially the platform companies, they are now move into a clinical biotechnology company because of this reason. Money is the later stage, and there is really disadvantages, because in the beginning, when you have the real innovation, people do not tend to invest in the early innovation because they do not see the money back, or it's, and this is very interesting from an innovation point of view, how we can get now in the early beginning that the real new stuff can come really to patients when there is no incentives anymore for investors to invest in early stage projects.
I can give you my perception of this area. It's just my definition. I think that, in Europe, we have a general problem. There's not enough capital on the market for the innovation that we have. This is when I compare the availability of venture capital in Europe with the United States and also with China, the other 2 regions, China and the United States, invest 5 to 10 times more per year in their companies and in their research, and especially in development. When I look at Europe, I think all the dollars, the majority of the euros, go into early stage research, not development. So there is a lot of grant funding, a lot of philanthropic approaches. You also mentioned Bill and Melinda Gates, for example, the foundation. These are all philanthropic approaches. So where they invest in companies in research organizations not to develop a drug up to the market, but to just produce new IP, new know-how, and not finalize projects. When I look at the later stages, so why there is more money, I mean, there's less risk, and the studies are just bigger, so you need to allocate more money. When I look at the stages in between, there are investors, but the risk profile is different. So you mentioned Merck, Pfizer, GlaxoSmithKline, so these are the big pharma companies. When they put the euro in these companies, the risk of them going extinct is almost zero. It exists. We can have an economic breakdown or something really terrible can happen in the world, and then these companies don't exist anymore. But usually they are set up to survive. When I look at early stage investments, for example, into startups, I know that every euro I invest in a startup company has a chance of success when we are in preclinical stage of maximum 5 to 10% or 1 to 3%, really depends where the technology is, but it's not higher than 10%. If I bet all my money on one company, it's a high risk of failure. When venture capitalists spread the money on at minimum 15 companies, they can narrow down the risk almost to zero. And usually when I look at the portfolios of venture capitalists, 1 or 2 companies repay the entire fund and leave enough profit to motivate their limited partners to reinvest in a second fund. So this model works really well, but the problem only arises when people bet their entire capital on one company, especially in drug discovery.
Yeah. Yeah, that's a huge problem, especially here in Europe, as you mentioned, that the size of the venture capital funds here in Europe are, in my opinion, too small. So when you look at the bigger funds in the Silicon Valley, so you see always a very, very excellent, I call it a very excellent expertise or expert department, what they always have. And this is what you complete. You very here in Europe. I don't want to see it's not there, but it's very limited because a specialist for CRISPR-Cas, for example, these guys are expensive, and the reason why I should join a VC is for European probably not completely clear. So he stays at the university or at a pharma company or at a biotech, for example, but we see a lot of people, especially in the genetics field, moving towards jobs in venture capital now. And they can then judge the investment. But this is very, very, very expensive to hire all these guys to have the expertise in your VC. And it only makes sense when you have very, very, very big VC, because then you have this 220 rule. So 2% is the management fee. But it's for example, billions, I don't know, 100, 200 billions of the whole volume, and it's in 2%. Then you can afford these people, and then you can make also better decisions. This is also what I see, that you have some kind of an advantage in your expertise in your VC when you have a bigger VC fund.
Mm-hmm. No, I completely agree to what you say. I think the 2 main points for me are, whenever I help scientists move the company forward, the success story is straightforward. You can found a company in Europe, you get a lot of grants and early stage support. So the minute a scientist has a breakthrough invention in a research organization, there are sufficient funds for the early stages. But the thing is, and you mentioned CRISPR, for example, and I think the story of Rodger Novak and CRISPR Therapeutics is an excellent example for how the process then continues after the foundation. So as far as I researched it, Rodger Novak and Emmanuel Charpentier met in Vienna and then decided to create a company in Switzerland and got early stage funding, obviously there, otherwise there wouldn't be a company. Then the next step was clearly to Boston. So to the United States, to fund the development in the later stages. And this, I think, is the big downside of Europe. We are good in early stages, but there are limited funds at the later stages. And what you said, the size of the fund, anything below 10 million euros in a fund doesn't make any sense. On one hand, you need the experts in the fund. On the other hand, you need to have enough capital to allocate in a big enough portfolio to have two winners at least in a pool of 10 to 20 companies. And for mid-stage fund, I think anything below 50 million. So mid-stage, preclinical, clinical phase 1, it's a little bit late early stage, up to mid-stages, is the minimum size of the fund, 50 million. And for later stage funds, 100 to 200 million. So it's these are the ranges.
So you mean the total volume or just per investment?
No, the total volume, the fund size,
This 50. I think it's still too less money. I think even 100. When you have 100, you can do probably 10, 15 investments. So it's always so that you have to keep some dry powder. For example, there's a recession, then it's difficult to find the next funding round. Then you have to put more money into your investments, otherwise they die. So I think in a fund with 10 to 50 million in a biotechnology space, in tech, I think it's different because you are faster to some kind of a service or product where you can show revenue, where you also find these investors that are more experienced with these e-commerce ideas, where you can raise then also bigger rounds and so on. But here in biotechnology, 10, 50 million is for a fund, in my opinion, like suicide. So you need really more.
I completely agree. It's the bare minimum to start the first fund. So when someone wants to start the first fund, it makes sense to get the first fund off the ground. It's even harder to raise money for a first-time fund, when the team comes together for the first time. So starting with a 10 million round to prove it works and that the team has a deal flow makes sense. But then up 50 million is the minimum. Is the minimum for the next fund to play in this game.
And it's actually extremely hard to raise a fund. When you start up your own VC, it's extremely hard because your investors in the fund, they want to see success. And how you want to prove success is that for this you need money to invest. It's really tough. It's really tough.
And it's still an open job for regulators. If any politician is watching, we need to motivate the institutional investors, banks, insurances, and also pension funds. It's a huge difference in the United States. Pension funds, for example, they are not allowed to provide venture capital. And when they would allocate also in Europe only 1 to 3% of their capital under management into venture capital, it would be paradise for drug development here in Europe.
But this is the general problem I see here in Europe, that I call it financial engineering, that this is not so common here in Europe. I don't know. So when we started the company here in Germany, it was extremely common that when there's a business angel, for example, that this business really wanted to have shares in your company, so you have to go to the notary. So, and the usual early tickets in this time 25,000 or 50,000 was too high, 25,000, but then you spend 1,000 or 2,000 just for your lawyers and the notary. And then 4 years ago or 5 years ago, then you had this convertible loan. So I don't know whether you heard about the Y Combinator convertible loan. So it's actually a template you can download from the Y Combinator. And this has really huge impact, because in the early beginning, when also it's not really clear what is the value, what is the valuation of your company, you just have this convertible note, and then this question can be answered then in the future when you make a real investment round, and there's then real numbers on the table, and you can say then to your early investors, for sure there's then this discount what you have agreed in your convertible note. So I'm a huge fan of this convertible note, and these are from the US originally. So
completely agree. I mean, big shout out for Y Combinator for the
yeah,
SAFE model. I started fundraising in 2006 in the industry, and there was no standard on the market how to get capital into a company. And sometimes I got offers of 100,000 euros for 90% of the company, which doesn't make any sense. And Y Combinator just issued, I think it's, I don't have the latest version in mind, but I think it's 7% for $125,000 in a convertible loan in a safe structure with which is really great. It sets a great standard for early stage investments for the founders and for those organizations who want to bet in early tickets.
Yeah. So actually, about valuation and in investment, it's very interesting. So, for example, last couple of weeks I heard from a friend. So he wants to raise 2 million, and then he offered some percentage of his company, and then he also talked to US investors, and the US investors were then offended and say, "You're not ambitious enough."
Yeah. It's a different world. It's a different world over there.
Yeah.
Seed rounds. The last two companies I had on the podcast who raised venture capital in Silicon Valley from NFX Ventures. The one did 7 plus 8. It's I think $15 million. He's located in Israel, but has a professional network in Palo Alto. And the other company, Samantha Dale Strasser with Pepper Bio, if I remember it right, I think it was also 7 million seed capital. Drug development, artificial intelligence. When I look at Europe, when we talk about seed capital, we talk about 200,000 to 300,000 equity, leveraged with 1 to 2 million public funding. So we are about 50% of the size that the seed rounds are in the United States. But in the United States, mostly it's venture capital, early stage venture capital.
Yeah, but I really have to say that, and keep it in mind, the US is more expensive. So I'm pretty sure if you have 2 or 3 million in Europe, you can go much more far than when you have $7 million in Boston, for example. So keep it in mind, because salaries are incredibly high in the Bay Area and in Boston. So and lawyers are also incredibly expensive in the US. So you see now some movements out of the US coming to Europe because they see the opportunity that the development is much more cheaper here in Europe.
Especially with the weak euro these days, it's also additional upside.
yeah. But this is when you hear about the salaries in Boston, for example, for a specialist, it's incredible. It's really incredible.
I see a lot of collaboration potential. I completely agree. I mean, bringing the early stage development to Europe, it's cheaper. There's a lot of expertise. There's a lot of public funds. There must be someone who motivates the US investors, because usually I learned that they get some tax breaks and tax benefits when they invest in US companies. There is not so much incentive to bring US capital to Europe. But from a business perspective, I completely agree. I mean, if they would like to, or if they would take the risk to invest in Europe, they could easily find an environment with less, where they could achieve more for each dollar due to weak euro and lower overall expenses in the early stages. And still in the late stages, then they have the hand on a company and can move then the asset and the team to Silicon Valley or to Boston and complete the later stage developments there, where there is more availability of capital. I mean, CRISPR Therapeutics, for me, is always the best example how you can move breakthrough technology forward. Let's go back to artificial intelligence. I started getting interested in digital solutions in 2013, 14, 15, but it was really challenging to talk with investors back in the day. The digital investors, as you mentioned before, were solely interested in quick-to-market products. Drug discovery is nothing near to that.
Yeah.
And the usual drug discovery, drug development VCs were still in their environment where they invested in molecules that tackle big problems and that can become the next game-changing technology. Artificial intelligence was in between. So it was not digital enough and drug discovery, and it was not drug discovery enough. Then suddenly, 5-6 years later, there is a big hype on the market. So I think every company that I get in touch with now presents an artificial intelligence solution. Where did this hype come from in the last years?
Yeah, the hype comes, in my opinion, that there's a lot of pressure in pharma R&D. So I used it before, the word erooms law, and this is a little bit like a Damocles sword in the whole industry. And I know that when you are a pharma executive and you meet your investors always ask, okay, your pharma R&D, your productivity is so low. How you can improve it? How you can improve it? And they ask every time how you can improve it. And it turned out that artificial intelligence was then a little bit like kind of an escape rule. So they say, oh, we do artificial intelligence. So we now can do all this on scale because artificial intelligence is this a synonym for investors that you have a scalable product, that your product is scalable, that your service is scalable. And this is now also adapted here in the industry. And you're completely right. So I see in talks now, every company is doing now artificial intelligence. So even they have done the same thing before. They coined it now artificial intelligence. So best example is molecular modeling, for example. So had this structural biology background because of my postdoc. We used a lot of molecular modeling, and in these days it was called molecular modeling, and nowadays it's called deep learning AI. The techniques, in my opinion, the algorithm were mostly or like a little bit the same. Now, of course, the algorithms are much more advanced for sure. But the data sets, and this is a success, my opinion, here, for example, when you look at Alphafold, Google Alphafold. So for every, I call it machine learning approach, you need a very good data set where you can learn from and you find the patterns. And Alphafold's success relies, in my opinion, more on the extremely well-curated data sets provided by an army of scientists all over the world. So there are, I guess, thousands of structural biologists. They solved crystal structure the last 30, 40 years, and they made them all public. So they are all publicly available. So you can go on the protein database. It's a website in the internet, and you can download 200,000 structures from there. And this is the curated data set they had to use. So and I would like to value this more than the algorithm that DeepMind developed, because the curated data set is often the big bottleneck in the whole development. And yeah, that's one point, that more and more data sets are becoming available. Here, when you look at the genomics area, we see a lot of good data sets now becoming available from the big biobanks, for example, the UK Biobank or Genomics England, for example. And these very, I call it excellent curated data sets are the basis now to apply these, I call it sophisticated or novel predictive algorithms.
It's a great point that you bring up, the different facets of artificial intelligence. When I look at the topic from the science fiction point of view, I always thought, wouldn't it be really nice that when a scientist has a new idea, that we have a machine where you can feed the idea into it, and the machine simulates the entire process from the first experiments up to clinical phase 3 trials. And based on this data, you can then get regulatory approval at the end of the day of a fully automated manufacturing unit. The drug just comes out and is ready to use for the patients. So this would be a dream world, so that you don't need this lengthy process in between, that you have supercomputers who calculate everything. What's the reality? What are the different facets currently of artificial intelligence and machine learning in drug development that are really reality?
So actually, so what you described, I don't think it's so much science fiction in my opinion.
Really?
So no. For example, when I look back at my postdoc, we had a very, very difficult protein structure to solve, but we had the luck that other structures were available with different conformation. And what I have learned is when you have a good databases, so we had plenty of structures available as a data, then molecular modeling makes more sense. And what you described, in my opinion, is not the problem of to have a supercomputer. I think it's more a problem of having the data. And when we come to a point where we have so much data available describing how to do to simulate in vitro experiment, to simulate clinical development and so on, then I think it's not so unrealistic what you describe here.
How can we get the data?
Yeah, we need more data. We need more, and very important is that it's curated, and we have to bring these kind of data to the public resource. I'm always an advocate to bring data to the public. What I see here, you have to make sure that in the early beginning, so when PhD students do their work in the lab, that everything controlled under specific conditions that makes it reproducible for others. And when you then have all these kind of results and feed this into your machine, then it makes sense. The other problem what we have still here in academic is that academic research is, in my opinion, still too biased, because the academic system works that I'm only got incentives when I'm voted by other scientists. So it's more clever to bet on topics where I know that other scientists are also interested in, because then they quote my work. So that's why we have these I call this clumps in specific areas. And actually this is a problem in the university and the research organizations to go to from these clusters of high quotations, of high citations, more into the other ways. And then what you describe is in my opinion, impossible.
These clusters are good and sometimes challenging at the same time. So since I'm not in academia, I can't comment on that. But I found the second point, which is more in my area of expertise, quite interesting. Did I understand it right? Accessibility of data is a huge problem. So that data that's produced in laboratories at research organizations and also in companies, in early stage companies, is not really accessible, so that you don't get access to the data to have a sufficient data base where you can start training your artificial intelligence on it. Is that the right understanding, or did I miss something?
So what we see is that a lot of data which is produced, I call it in these smaller units, which can be just a research lab and a university and so on. They're very free in what they do and how they conduct their research. So that's the reason why it comes that when you want to repeat the experiment in your lab, then you cannot come to the same results, because I don't know, there are some other conditions that are actually not described in the protocol. But because for them it's, I don't know, probably the air condition is always on 20 degrees and your air condition is constantly on 25 degrees, and these 5 degrees, they makes it, so I don't know. But in the early point, I agree with you, there's a lot of bias in the research, and it's very difficult that we destroy the bias in the research in the early phase, and to make this research and also available. because most of them, most of these results are, in my opinion, unsuccessful. And unsuccessful results are not interested. It's not cool for a scientist to publish unsuccessful results. So there's no incentives for him. So he will not see citation for this work. But here, I guess it's a huge potential, in my opinion, that machine can learn what we can do and what we cannot do.
Yeah, that's a big question, I think, also in companies. I mean, when after raising funds from venture capitalists, sometimes the initial hypothesis doesn't simply work and the company is basically shut down. What happens afterward with the data is quite simple. It stays in a corporate shell. So no scientist has any incentive to publish the data in a publication. And also the VCs don't have an incentive to open the data room to the public. So it just sticks around in a corporate shell very often, in my opinion, where sometimes when they're lucky, they just merge it with another portfolio company. Then it's open for that portfolio company, but a lot of companies just don't move forward. No access.
Yeah, and the data is completely messy. So it takes ages to curate the data. This is also one point. And nobody is interested in invest time in something where they really don't know whether there's a reward for them.
Do you have any ideas, or are there any ideas on the market that you are aware of how to solve that problem?
Actually, yeah, we are talking about early research, and I have actually no idea. What I see is the biobank data, for example, when you use these big cohorts. Here you work with thousands of participants, for example, and they are really, really well curated, the data. And this brings really standard to it. And what we see with the biobank data and also with our customers who have then always their lab experiments somewhere from an academic partner or from the academic founder, for example, you can test these in vitro data in the population data, and then sometimes it turned out that the in vitro data was not done properly, or it's actually wrong, and we cannot see it in the population data. But we see then in the population data also the signal, which was already postulated, and then it's really good, and we can go on. So to answer your question, I think we do not have the resources to clean up all the early stuff, but we can use the data we have from the later stage, from population-based, from clinics, from hospitals, and so on. And here we can prove at least the assumption from the beginning. Very, very good, in my opinion.
No, it would be really great to get access to all the data. So basically you need a super curator who gets all the data and structures it in a way that it's usable for later stage.
Yeah, but we see now that the biobanks, I refer always to the UK Biobank because these guys, this is really fantastic what they do. And I'm a little bit disappointed that we do not have it here in continental Europe, especially here in Germany or in German-speaking countries, because I see that this is a kind of an infrastructure we really need for the future. So people stick too much on the research and on the data, but when they look back to the pandemic, there was then the so-called British variant, the British mutation, and this actually based on the fact that only the British had the capacities to sequence enough COVID tests. And this is just based on that they had the infrastructure for the UK Biobank. So UK Biobank, they sequence 500,000 people. So when you want to sequence 500,000 people, you need the machines to sequence. And if then a pandemic, then it's for you easier to sequence also the COVID tests to see whether there's a new variant or not. And in my opinion, the UK is extremely benefiting now from the infrastructure, UK Biobank, in two ways. They have now, I call it a proper industry in sequencing and DNA sequencing, but they also have now a lot of data companies strong data companies in the area, and more with the time you will see that they will take the data and will develop products out of the data based on the UK Biobank or Genomics England.
I was not aware of that. I thought every country uses the data properly. That, I mean, we also in Austria had a lot of tests, a lot of testing. It was free for the population, which is very good. But do we really don't have the necessary infrastructure to interpret the data afterward and to use the data afterward in research, like the example you brought up from the United Kingdom?
You mean with COVID?
With COVID, for example, I mean, it's just
yeah, COVID. So the British, they were much more closer to the pandemic than we were in the time. So you have your participants in the UK Biobank, and they are frequently monitored, the participants, and then you know already that whether they have been infected by COVID or not. And then they conducted antibody tests, for example, to see how many people in the whole UK Biobank have antibodies. And then you have an idea how many people have been already infected by COVID-19 in the UK or not. Then, of course, when they come, the monitor picking the participants, so officially participants of the UK Biobank, come to their physicians and then say, I'm feeling sick. And then a COVID test is done. Then this is also going to the UK Biobank facility, and then they can sequence. We just only do a PCR. We look, is this positive or negative? But they have then really tested or sequenced what kind of variant we have here. So they were much more closer to the pandemic. And when you're closer to the pandemic, then it's much more easier to say, okay, it's worth to shut down here, for example, or what kind of medication is necessary. So I see here a big disadvantage in Germany, that we do not have this biobank infrastructure as the UK has.
Yeah, would be a great thing. You named your third post about artificial intelligence in drug discovery and development. Why the way we do science does not work for drug development, and this seems to be one problem.
Yeah.
I mean, I personally would not care if anybody uses my data for drug discovery purposes. So, for example, whenever I'm tested, or genetics data, or also, I mean, I have Apple Watch, and that tracks every time when I run, and I believe also Apple is investing a lot of capital into improving data readouts from human beings. So if I understood you right, your perfect world would be for moving drug development forward, that all this data that comes from human beings is structured into one huge database, then can scientists use for their predictions. Is this the simplified right understanding?
Yeah, it's simplified, but I would like to say that when you look at the UK Biobank has so sick people are overrepresented. I would really want to point it out here, and this is important. So if you're healthy, then there is actually no need to monitor you. But if you're sick and there's no treatment available for you, then it's good that you donate your data to the organization and to the infrastructure that the research facilities and also biotech pharma companies can use the data to test in silico ideas in a very, very early stage. And this is really what I would like to mention. So when this data is available and you can test your hypothesis in a very early stage, this brings so much. This is what people cannot think about. It's so important. It's it's the earliest time in your development to have an idea whether it's work or not. And if you can prove it then in the data, then you have also more idea about the endpoints for your clinical trial, and then it's then much more easier to set up a clinical trial, to find a cure, find some kind of treatment, and it is so cheap compared to the standard way, so that even you have a rare disease and probably only 10 or 20 guys in the world have this disease, it makes it so cheap that there's still a realistic chance that this clinical trial can be funded, because the endpoints are so clear and the whole model is so good that you can go on with for example, publicly funded clinical trial to get a cure or at least a treatment for your disease.
I couldn't agree more. There is an interesting point in what you said. You said that we only have data from sick people. Wouldn't it be nice, let's phrase it differently, the question, what would change if we had access to all data, not only sick people, but also the healthy population? Would that be an improvement to drug development processes?
So this is a good question, and I would like to call it in this way, no.
No?
No. So because the dimension there, it's incredible. So for example, we differ in 200 million positions. So it's so-called single nucleotide polymorphisms.
Mm-hmm.
So in 200 million positions, my gene differs from your genome. So in overall, it's 3.5 billion DNA letters, but only in 200 million positions we differ. So when we look for interaction in the data, sometimes it's so that you need 2 gene variants to have the disease. So it's in 200 million multiplied by 200 million. And then you come to a space which is so big, so there are not enough patients on this planet. So this 200 million multiplied by 200 million means that you have to sequence 5 million times the human population.
Oh,
so
okay.
So to get to see the statistical power. So I always say that, and this is, it's not cynical. So it's just only a very scientific, a very rational conclusion is that we do not have enough sick people to conduct proper statistics with the data. Because the dimension, and you cannot imagine, is because this is a space. So 5 million times the human population we need to cover, to understand from a statistical point of view, the human genome. So this is huge. And we are only talking about a 2-way interaction. If you're talking about a 3-way interaction, it would be 200 million multiplied by 200 million multiplied by 200 million. So it's like, it's unbelievable for us.
Yeah, so, I was always bugging my doctors with one question. Yeah. When I had my yearly checkup, usually got the answer, you are healthy, so come back when you are sick, which is a good thing. Yeah. But then I always asked my favorite question. I heard that people get cancer when they are in their 40s or 50s. What can I do now? So 20, 30 years ago, when I was in my 20s, now I'm now 48. What can I do now to avoid getting cancer? So, and to that, there was no answer. And was hoping that artificial intelligence could solve this problem, but...
Yeah, so some years ago, so we had here a project. We developed prediction models for breast cancer and for type 2 diabetes, and also for Alzheimer's disease. So one of our first projects was to develop a prediction model for Alzheimer's disease. And there's this famous gene, ApoE4. It's when you have it, then it's very likely that at some age you will have Alzheimer's disease, but this can be with 60, but it can be also with 90. So it's difficult. So, and it was a very nice, was a good example for what we call explanatory statistics, because it is extremely overrepresented in Alzheimer patients, but it has actually no value for prediction. So because it's for you important to know when you have it. So when you have Alzheimer's disease, when you are 90 years old, and you will only, when you're 2 weeks or 3 weeks before you die, then this is actually not really of value for you. The value is when you have it, when you're 50, something like this, and how you can prevent in the outbreak of Alzheimer's disease. And we had then a prediction model, so we had then found interaction of ApoE4 with other genes, what provided us a better, or what enables us to predict in a better way Alzheimer's disease. But here was then the problem, okay, now you know that the likelihood is much more higher, but there's no treatment. So it's unethically to give you the information that you will have Alzheimer's disease when you're 50. So so we have done this for a pharma customer, but you see, this is the prediction. I know is a lot of, I call it a myth a little bit in digital health, that prediction is actually something that has value in our health system, because first of all, when there is no treatment available, then it's unethically. So a physician should not tell you this. And the other way around is what is the accuracy of your prediction? Because there's then still a chance that this is just nonsense. So you really have to prove it. And to prove it means that you have a prospective clinical trial over the next 20, 30 years. And then it's very expensive to prove it. Very expensive, and it takes ages to prove it.
I mean,
so
a solution or a therapy can always be a product, like in the pharma industry. So it's usually a pill or it's a liquid that helps patients. For me, I mean, this is one of the driving questions of my life. I would like to understand how diseases develop, and a therapy then also can be some lifestyle changes to avoid getting a disease. Or, I mean, the ideal point for me would be live happy and healthy for 7, 8, or 9 decades on the planet, and then when life comes to an end, die fast. So to avoid this long time of a disease, or suffering from a severe disease for 2 or 3 decades. And for that, I think it's, in my opinion, it's necessary to understand how does the biological system come from a healthy state up to a very long progressing state of disease, instead of just shutting off from one day to the other. And I was hoping that with artificial intelligence, it's easier to get a better understanding beyond drug discovery and drug development.
Yeah, definitely. So, for example, type 2 diabetes, we have extremely good prediction models now. So I think it's 99% accuracy you see in prediction model for type 2 diabetes. So there's a lot of, we know that there's a lot of genetic impact, and then it's actually your body weight you see in a direct relation to it. So if you have problems with your body weight and you do not do some sports, and then you have this genetic impact, and it's very likely that you have it, that you will develop type 2 diabetes. Coming to your question, so this reminds me to a project we also had. So we looked in the genomes of long living people, so 100, 120. So they checked them in their genomes and compared to people who died early. And there was always the idea that there is the longevity gene, but actually what we saw, there is no longevity gene. There are I call it shortgevity genes. So there are really genes that are known for diseases, and if you die early, these genes are extremely overrepresented in this cohorts compared to the long living people. And this is actually a little bit my takeaway. So I think at some point with your education, you have an idea how to live healthy. But if you have just a normal genetic makeup, then there are actually no big risks for your life.
It's very interesting to talk about the different facets of artificial intelligence. When I asked the first time these longevity questions out of personal interest, in the 90s, there was almost no answer from science. So I think it's just really way back 3-4 decades. And when I look now on the internet, I think the availability of data is much better. There was some years ago, 10 or 15 years ago, I read a study from, I think it was Harvard University, where they did a study over more decades, where they observed people with a questionnaire, yearly asked the question, what are your lifestyle habits? And then came to the conclusion that there are 4 or 5 factors that influence health tremendously. And now with artificial intelligence, we could scale it up. Is there any chance to get the necessary data to get more and more insights into these mechanisms? Or, I mean, in Europe, we have our GDPR rules, which protects the data. What are the limitations of regulations that limits the development of artificial intelligence in drug discovery on one hand, but also on the other hand, in getting more insights and understanding of the development of diseases?
Yeah, there are two things. So talking about the GDPR, I think it's just an excuse for bad research. Sorry, it's very harsh the answer now, because it's actually research. So when you look into the legal text, you see that when it's research, then GDPR it's not so important. So you can use the data. What I always see is people conduct here clinical trials or genome-wide association studies here in Germany. And when you look a little more closer, then you see the work is not properly done. But it can be revealed easily when you see the data. But they then say, no, there is this data protection issue. That's why they do not disclose the data. So they keep a little bit the fact that they have not done it properly. So this is a little bit my opinion on this, because GDPR, people talk about this. When it is research, then it is allowed when you look really in the legal text. So this is really actually not a problem. To your other point with the longevity, there's a lot of movement now in the scene. And there is this famous phrase, I can also now cite here. So when you look at all these Silicon Valley billionaires now, so when they were young, they wanted to be rich. When they're rich, they wanted to be young. And that's why you see now a lot of money from people, for example, Elon Musk. They heavily invest in longevity. It's really incredible, and you see a lot of things now upcoming, in my opinion, because for them money is not an issue. They have a cool life and they want to stay alive. They want to enjoy their cool life. That's why they are there, and they have actually unlimited resources from a money perspective. That's why I'm pretty sure that we see a lot now, and a lot of data will come. And I'm pretty sure they will also put this data publicly, because I haven't mentioned this before, but this is really important. So if your data is in your silo, then the use of the data is limited by yourself. But when you put the data into the public, then you really see that other people come with new ideas, with new methods, with new technologies, and they really make it useful.
Yeah, that's true.
And they also find the mistake in your data. When people always say, that's only mine, and it's the GDPR, whatever, then you can make sure that there's something wrong with the data. Because when the data is proper, it's curated and so on, you invested so much, and you personally want you want to see the result out of the data, then you're more open to say, okay, let's make it public to all the people outside of the world, because then we can have all these kind of check and balances from other sides that there are good results, and we can really use it. And in the end, I want to live longer, more healthy. This is my actually goal. And as I said, from the Silicon Valley billionaires, so they do not argue with money. They only argue about their life and about their lives. And then,
yeah, this was also more of selfish interest in the 90s. My big questions: I want to stay healthy. I don't want to get sick. So what can I do to increase these chances of staying healthy longer? You never can avoid any accident. So, I mean, sometimes life happens, but I think also lifestyle has a huge impact. And I'm curious to hear also from people like Elon Musk and Bill Gates, and I think also Tim Cook, Apple might be something doing in that area. What conclusions they draw when they put really big numbers behind it. I think Christian Angermayer is also someone who is investing heavily in that space. When we look at the situation right now that you mentioned with the availability of data, wouldn't it be helpful to get access also on what you call bad data, or data from studies that failed, to increase the know-how?
Yeah, so I mean, bad data is, I call it badly curated data.
Okay.
So for example, when we started companies and we had only access to, I call it shitty data sets. And then we had to build our own quality control unit. And sometimes it turned out that 20% of the data was useless. So because really standard quality control, this data could not pass it. Good example was then, so you have the genomic information, and then you have the phenotype, and the phenotype says, so this is a woman. You look in your genomic information, you see it's actually a man. So then there was then the discussion, yeah, probably it's intersexuality, but 20%, come on, nobody can tell me that this that happens 20% of the time. So this is really huge. And this is just only a sign that it was not properly done. But anyway, so what we have done here at Biotx is that we took these badly curated data and we curated in our own ways after standard our standard quality control. And from this data set, so roughly 80% were then is useless. Is useful. Sorry, is useless. Is useful. 20% is useless, and 80% is useful. And we can then go on.
So to improve drug development, if I sum up the conversation up until now, in early stage development, there are two problems to solve. One is getting access to data, and the second one is the curation of the data. And companies like yours, then when they really sit down and do the second step and they curate the data, you have 80% of the data you get that is useful for further creation of solutions.
In the beginning, but what we see now is that when you look at the biobank data, which is coming more and more available over the time, this is now really excellent. So they have proper quality control units and so on. So in the beginning, it was mostly useless, but they have really learned. So for example, the Wellcome Trust has invested massively in the beginning in this area. So it's the Wellcome Case Control Consortium, for example. And this data, even the results were published in Nature. So there's a lot of bad quality in it. But the Wellcome Trust, so they paid it. But they also said, okay, look, what are the findings now 5-10 years later, what we have done wrong, and how we can avoid in the future. And these learnings were taken especially by UK Biobank and Genomics England, so in the UK, and then it's used in this normal biobank data. So I think we are here on a really good way. So I know people always complain about the quality data, but in my opinion, nowadays, it's a little bit also a kind of we call it bike shedding discussion, because people do not understand the algorithms and how you can provide value and so on. They always say, oh, the data is shitty, you know?
I mean, for me as an economist, quality is a function of capital. So, I mean, you mentioned before that studies are cheaper in Europe, but when we have less capital, we can't do as much as deeper pockets can achieve. And also Wellcome Trust, I mean, I completely agree. Kudos to Wellcome Trust. They're doing a lot of good things and bring research and move research and development forward. You mentioned biobank. I'm just curious, maybe it's a stupid question, but I ask it anyways. In your opinion, to get more data that moves AI drug development forward, how many biobanks would we need in the world to increase the quality and the accessibility of data?
We need only one biobank, a world biobank.
But with how much capital and how much resources to get all the data?
In my opinion, when you look, for example, at the Western at the development countries, for example, Western Europe, in my opinion, we are talking about, then let's say it peanuts, because a lot of what you now do is done by the daily routine in the hospitals. The only way is now to document it. And put on top, for example, genotype testing. So genotype testing, it costs 25 euros. 25 euros is genotype testing. So it's when you run these genotype chips from Illumina, for example. So it costs you also with the quality control and with in bioinformatic procession. It took you, or it takes you 25 euros. So it's incredibly cheap. And a full human genome sequencing is now below 1,000, and last time I read 100 US dollars. So this is so incredibly cheap now. The people who cover the data, this is the physicians, the nurses in the hospital. And I think it's only important to keep an eye on sick people, because there are enough healthy people already available where we have the information as a standard control. And then you can manage this. So in my opinion, there are just only excuses with GDPR and that there's not enough money available. So what you can save, what you can really bring to the people and to the patients, I think it's incredible. But the investment, in my opinion, and this we can call it here with the concept of the entrepreneurial state. It's almost nothing, because they pay already for this.
So we have the data, we just need to structure the processes properly so that the data is curated and made accessible for scientists for further research.
And this, and in my opinion, and this is my learnings from the last 5 years, when you bring the data to the public or make it public, then you also see that a lot of physicians probably don't do their jobs properly. So you have then also kind of a quality control, how people, because you see everything, and then you see that the person is diagnosed, for example, for a specific disease. But when you see then all these biochemical parameters in the data, then you see, okay, it cannot be this disease. And I think here there's a lot of there's a real problem. I mean, that physicians, they do not want to be monitored by this in this way. And I think this is actually more the problem than GDPR, or that it's so expensive to set up a big biobank here, especially in Europe, because we pay already for this, and we pay enough for this, and it can be covered easily because it's not so expensive.
No, I completely agree. And I think the healthcare system in the United States and in Europe is far too expensive anyway. So it's unfinanceable in the state we have it currently. And when I just sum up and in my mind and try to look more at it from a science fiction point of view again, everything that we can do that improves diagnostics, for example, to diagnose diseases much earlier, or speed up drug development processes that bring drugs to the market much quicker to cure diseases, and also understand on the longevity point of view. I think this research is also very valuable, because it helps people to understand how they can stay healthy much longer. It drives down the expenses for the healthcare system. And this is, I think, one of the big problems that Europe and the United States currently have. And artificial intelligence plays a huge role in that.
Yeah, but here you see a huge difference in the population, in my opinion, because you have these well-educated people. They care about their health, and they are willing to invest in their healthy bodies. And then on the other hand, you still see a lot of not so well-educated people. They just only say, okay, I still want to drink my beer in the pub, and I still want to enjoy my life in this unhealthy lifestyle. But just deliver me the pill. I take the pill every day, and that's it, you know. But I think we should talk a little bit more about the genome. Sorry,
Sure.
to point it out here, because I see now. And I think this is the origin here why we talk. So the human genome. So it was sequenced more than 20 years ago. So in roughly 2000, 2002, it was then published. But we still now see more and more impact of the human genome. So the pharma industry. So as I said, erooms law, for example, there's now breaking erooms law from 2010 and so on. And I see here the real value of, and I actually dislike the word artificial intelligence because there is no definition of intelligence. So how you want to define artificial intelligence. So I would like to call it predictive statistics and also causal statistics, causal inference. So, and this is actually the huge opportunity we see now. Because, as I said before, the big problem is to show efficacy of your drug. And when you look from a mass perspective to it, so efficacy and causality is actually the same. So in 2019, there was the Nobel Prize in economic science for Esther Duflo, for example, and she had this brilliant idea of conducting randomized trials to get an idea whether the development aid really works in India or not. So she conducted prospective trials. So she selected in a randomized allocation a group of poor people in India who receive some development aid and the others do not. And then she could compare it. And this is actually the gold standard they took from medicine to the economic science. But this is so expensive. This is really so expensive to conduct a clinical trial. And this is also, we had in the beginning of our conversation, clinical trials. This is then where you have to find the patients and you have always not enough patients. Recruitment is slow. Then you have, so your assumption hits the reality, I call it, that some of your inclusion criteria, they do not match with available patients and so on. And in economic science, and it was very interesting that 2 years later, so last year, there was a Nobel Prize for causal inference. And so you have the problem in economic science that you cannot run trials, prospective trials all the time. So they looked in ways how they can use observational data and prove causality out of observational data. And then they open up the completely new division of statistics, and this is causal inference. And to point it out, I see here now the biggest issue here for artificial intelligence, for machine learning, causal inference, because when we then bring causal inference, so we just only check for causality in the data, and this is often via this, I call it the 3-point way. So you have your instruments, that's actually your genetics, and then you look for a trait and for the disease. So very famous is cholesterol, for example. So you have genes that can increase cholesterol in the human body, but on the same way, they increase the risk of heart attacks. And then you now can model, if you have genes that just only increase a little bit the cholesterol, they only increase a little bit heart attacks. If they increase a lot cholesterol, they should also have an effect a lot on heart attacks. And this relationship you can then model, and then you can say, okay, this is causal. This is the causal relationship. And to apply this now here, combine this with predictive modeling. So you have prediction models where you say, okay, I take now the point where I know this is actually, or I think it's causal, and I took it out of my model, and then I look for my prediction of my model. So here I see a lot of potential. And the genome gives you the link to the biochemical level, because you actually see whether you have this alteration in the genes or not. And if you have them with the biobank, 2 million, for example, genomes fully curated and with phenotypes, then you can mimic actually whether some alteration or some modulation of a specific drug target gene can work or not. And here I see a lot of potential.
Can we say mimic? Does mimic mean that this study is simulated?
Yeah. So
not because I mean, I just remember the conversation you had with Emily Mitchell about decentralized clinical trials. You mentioned the problem that getting enough patients, large number of patients into a study is a science by itself. So it's not that easy to do a 40,000 patient study in a phase 3 setting like Moderna did. Even getting 200 patients sometimes in rare diseases is quite tricky and difficult. And I saw some cases where it can take 5 to 10 years to get this patient population that really is willing to participate in a study. And one solution is decentralized clinical trials. And did I really understand you right that we are talking about with artificial intelligence about simulating entire trials in a computer?
So what I would like to point out here is that you have these two things when you want to develop a drug. One is you need a ligand and you need a safe ligand. And in my opinion, there are a lot of methods now available where you can test the ligand in forehand and see whether it's safe or not. And the other big problem remains is the efficacy. So do you see an effect due to the modulation of the target you choose? But what you can do now is you look at the genome of, for example, what we do is 2 million people, and then you look for alterations for the specific gene. So we have done this with COVID in the past. So we were interested by people have a severe COVID course and the others just only have mild moderate. And we applied then this causal inference methodology on these two cohorts. And what we figured out is that severe COVID-19 patients had higher neutrophils, much more higher neutrophils than people with mild moderate cases. Mild moderate COVID-19 cases. And we could really demonstrate this with prospective regression modeling, drop-one analysis, propensity score. So with a lot of mathematical methods, we were able to show that there's a strong evidence for the neutrophils. And then we went on to look into the genetics of the neutrophils, and then we saw that genes that have been significant for neutrophils, for the number, also for neutrophil cell count in general. And here you can then use the entire population, 500,000 people, and then you also see smaller signals because the population is much more stronger, it's much more bigger. So even you see then effect sizes that are smaller than usual. That's why when you look just only at COVID-19 patients, so in the literature, there are only 1,000 or 5,000 cases available. And these populations are then still too small to catch up all these very interesting genetic variants. And this was this kind of trick that these, you can say, artificial intelligence methodologies, they enabled us to find a causal trait. This was the neutrophils. And that then we can look in the entire population and look up what kind of genes modulate neutrophil cell count. And we ended up with CDK6. So CDK6 is an enzyme which is responsible for the cell cycles originally, but has an extremely strong impact in neutrophils, and especially in a specific job that the neutrophils do as part of the innate immune response. And this is so-called netosis. So this is a special form of apoptosis. And these neutrophils, they just blow off and they release so-called neutrophil extracellular traps. And what we figured then out is that all these drugs that have been tested successfully so far today, so bamidicitinib, for example, this is a Janus kinase, was found, I think, in 2020. It's a Eli Lilly compound, and then tested successfully against severe COVID-19 patients. And then another cancer drug so far was recently also tested successfully. And what we could then show in vitro experiments with a partner from the University of Bristol is that all these compounds, they inhibit actually these net neutrophil axillary trap formation, so netosis. So what I want to tell you here is that it was just a simulation, but when you do it now just with the population data you have, you're so close to a clinical trial. So that's why it makes sense to simulate it first, using the naturally occurring alteration in your genome to see whether there are impact on specific traits, which then are causal for a specific disease. And
so your model was able to rightfully predict what's going on in the human body, where the big problem is. And all the therapies that came later to the market then basically tackled exactly this problem that you predicted.
Yeah, you can say the drugs, and this is now what is now in revision for publication. We could see in our analysis that all these kind of drugs which have an impact on the severe COVID-19 status, they have an impact on the specific form of apoptosis on the mitosis. So they inhibit this overreaction of the human body, and the overreaction is mostly, you can really narrow it down to these neutrophils. And so this is what we could see in the data. Yeah.
So when I would try to think in commercial applications, so one application would be that you have a tool at hand for the pharma companies that before they go into clinical development, that they check their compound, their asset, whether it really tackles the problem that you predict, and then include it in their decision-making process, whether it makes sense to move with that compound forward, or it's better to switch to another compound.
Yeah.
Would it be a possible commercial application of your development?
This is actually what we do. So we offer this in a service model. Mostly we call these emerging biopharmaceutical companies. So it has a little bit the reason that big pharma companies, they have their own departments and they think they can do this better. But anyway, no comment from my side. So and actually the reality is actually the sales cycle with the big pharma companies. It's take you 1 year or longer. So we're working mostly with smaller companies, and then the sales cycle is much more faster, which is for us more important. And what we do with our, we call it synthetic clinical trial, that we provide not only an idea whether there is efficacy. So this is what we have done here with COVID, for example, but what you can also check is when you have a molecule already in the market and you're interested in a new indication, for example, especially in the rare diseases, because the data is there. And as I said, even with a small patient group, we can determine the causal traits, and then we can go back to the bigger population, and then I call it simulate, because on these disease patterns, the patients in the big population, and look whether there is an effect on specific genes or not, where your original molecule, we know that they can modulate these traits or not. And the last one, and I think it's also interesting, I said before, safety can be checked in the early phase. That's right. For some in animal models, for example, but due to the fact that you only test the compounds only for a specific period of time in the beginning, it's just only you want to move on. So you do the test for 1 or 2 months, for example. But sometimes when you have a treatment which has to be given or has to be administered for years, for example, for chronic diseases, especially, then you really have to check whether the inhibition, or mostly it's an inhibition, or the modulation due to your molecule to a specific target does not induce another disease. For example, in the past, we saw that the inhibition of a specific channel could lead to epilepsy, for example. But this was then is then really a danger. And but you have to know it before, because you then can think about how long the treatment can be, so that you say, okay, this is just only for the indication. So this can only be done, can only be given for a specific period of time, and then we have to check the patient whether there are symptoms regarding in this area or not, or are there, for example, genetic variations which makes the effect even stronger, and we have some more evidence, more chances, higher likelihood that this adverse effect will occur. So these are the three things you can do actually now with the data. So we check for the efficacy, you can check for new indication, and you can check for target mediated toxicity.
That's awesome. So drug development simulation, I think, isn't one problem also in clinical trials to find the right patients. I mean, you mentioned COVID, and I was thinking, while we were speaking, I was thinking about this problem. So I mean, from the information I have, which might not be right, but my perception of the COVID vaccines is that they work really well for severe cases, and don't have much effect when people are not at risk to get a severe disease, compared to the situation when they just sit at home and get over it in a couple of days. So from the patient perspective, but also in drug development, I think the interesting thing for trials with vaccines then would be to find the patients that are highest at risk for getting the severe disease, to also see the effect in a clinical trial setting. So whether it works or not, the vaccines might not be the best example because for the dynamics of vaccines to protect the healthy population. But when I think cancer, for example, does your solution also help to find the right patients for a study? I think just when we bet a lot of capital in a clinical trial and we take the wrong patient population, and theoretically the drug would work very well in a different kind of population, then we actually include it in a trial. When such a clinical trial fails because of selecting the wrong patient population, I think it's really hard to bring it back. So can your solution help also in that cases?
Yeah, so we had another project here in the company. It's lactoferrin for a rare disease. The very rare disease is called Ligand disease. So it's an autoinflammation disease, and you have these painful problems with your eyes. And the overall idea when we were introduced to the compound was that in the Ligand disease patients, iron would be missing. So lactoferrin, and you heard ferrin, so lactoferrin is able to bind iron. So they said that this would be then an iron thing in Ligand disease patients. So that was originally claimed. And then we looked a little more closer, and we run our causal modeling on this, and then we figured out it's actually based on eosinophiles. So it's a specific you can say immune cells that are going into the eyes, and then they start an infiltration, and the infiltration is then the cause of the vasculitis, which is then the real cause of the Ligand disease. So what we could figure out is that the eosinophiles is actually the point. It's a problem, and then you can use the eosinophiles as an endpoint for your clinical trial instead of the iron. So with the iron, because the iron has actually no clue in our causal model what we see in a big population and so on. So when you now go into a clinical trial, you just look for the eosinophiles instead of the iron, and then you have a better endpoint because you have already proven that there's causality between the eosinophiles and the outcome of the disease and not of the iron. When you have just the assumption still with your model with the iron, then the iron you see everywhere, so to say it in this way, and then it's very likely that you fail, because you have just looked for the wrong parameters in your data. And this is really a strong thing, because when you can choose better endpoints, you can also, and this is a biostatistical problem, you then also can select, or you can choose fewer number of patients for a clinical trial, because what you want to see is much more clear for your and then you only need so much patients anymore as in the beginning. It's a smaller group, but still, if you show this effect in the smaller one, the p-values you will get should be still significant. And this is, in my opinion, the real impact, because finding patients, this is the next big thing after you predict the efficacy in genome-wide association data and population data. So you still have to prove it prospectively for the regulators. But if you, for example, for a rare disease, you have to recruit 1,000 patients. So this is like, okay, forget it. You will never find 1,000 patients, even you have sites all over the world, and you are willing to pay incentives for patients to join. It's really tough. And here's also another point we really have to see when it's about this causal AI thing, and are synthetic control arms. And synthetic control arms, they are a fantastic thing. I don't know whether you heard about this.
No, not yet. What is it?
So the origin is in oncology. And oncology is so that for some tumor diseases, there were some drugs available. I call it in this way, very, very simple. So when you now have a new, and you want to come with a new drug, you think personally this is a much more better drug to the market, you have to conduct a clinical trial. And it's so that in the standard setting, you have these placebo-controlled randomized clinical trial. So you have your population, and some will receive just a placebo, and the others will receive your new drug molecule. But to receive a placebo for a tumor patient means actually his death. So, and then you run an unethical clinical trial, because half of your population will die for sure, because they're not treated. And actually there is a poor treatment available. I call it a poor or not so good treatment available. But if you don't give them to them, then this is really a point, you know. Then it's the idea, okay, I do the clinical trial against the standard of care, and this is a poor treatment. And then they had sometimes the problem that these poor treatments, they are off-label, so they are not really officially given, and so on. There was not, so it become more and more problematic. And then you had the possibility that you took retrospective data of tumor patients, and you just model now the progression of the tumor growth. And this can be really done in a simple Excel sheet. So it's not so much sophisticated. Turn to be honest, it's very simple, but you need the data of the tumor patients, and you simulate in the way how the tumor is growing without the treatment. And you just only have one arm of real patients, and these real patients receive your medication, and you test against the synthetic clinical control arm. It's it is established, I think, last 5 to 10 years, mostly in oncology. And these synthetic clinical control arms, so this is also a causal inference methodology, developed actually also here from the one guy who received the Nobel Prize last year, because he wanted to prove that immigration from Cuba to Miami does not have an impact on the unemployment rate in Miami. But then he wanted to compare Miami with another US city, and then he realized there's actually no. And so then he created the synthetic clinical control arm that he said, okay, so this is now Miami without the immigrants. And this is now also coming back to clinical trials. So this idea of causality, and I see here also now a huge improvement. So a lot of things now going on that you can decrease the number of patients in the clinical trials. So, and when people know that there is no placebo-controlled randomized clinical trial, so there is not a chance to just receive the placebo, because this is actually also some kind of jeopardy for these guys. Then they are also more willing to say, okay, there's a new treatment, and I read your papers, and it looks good for me, and there's really a chance that I can be healthy after the clinical trial, then they go for it. Otherwise, people say, okay, when there's a 50-50 chance. We saw in the past.
But what about the bias? I mean, wasn't the reason to invent double-blinded clinical trials with placebo control to reduce bias? Is that a problem then? I mean, when people know already that there will not be a placebo, does that jeopardize the results of the study, in your opinion?
Yeah, but you have to done them as a synthetic clinical trial. You have to done this then properly. So here you really should then see the impact. But I would like to tell you one little thing about the thing we have seen in the past in clinical trials because of the jeopardy for the patients. So in the past, it was then more and more difficult to run these clinical trials in Germany because people say, okay, then there's a 50-50 chance that I got them actually nothing, then I will not participate. And we saw then a lot of movement of these clinical trials and towards Ukraine and Georgia, for example, because there are so poor health systems that patients say, okay, so the standard of care is nothing. So I participate in the clinical trial and then I have the 50% chance that I get the treatment. So this is what we have seen in the past. And that's why I see these novel statistical methods. There's I call it a trickle down effect, and we know it in economics. Here also for patients in lower income countries.
Before we come to the last two questions, I want to check back if I understood you right. You said for simulation of synthetic control arms, Excel is enough. At one point, is it really Excel? I mean, Microsoft, the wonder tool that we have at hand.
So in the beginning, it was really Excel. I know that the big providers of synthetic clinical trials and will not call names here. They will say they also use sophisticated algorithm, but in the end, you can also do machine learning with Excel. This is, I know a lot of people will probably it will end up now with a shitstorm, but it's possible. You can do it, and believe me, bigger organizations, Microsoft Excel is still the standard.
Yeah, I know it also from other areas, but my experience is 20 years ago. I wanted to just check back if I have still the right understanding. It was in information technology. It was not in the pharma industry. But very often when there was a salesperson saying it's an automated process and the data is handed over automated, the reality was that someone freelancer was sitting somewhere in the world, taking the data from one database, transforming it manually in Excel and uploading it to the next database. So this was very often in the reality. And it's good to hear that Excel is still useful today. So Bill Gates did a good job at the end of the day.
The principal math, what you can change, So,
mathematics don't change.
When the data is becoming bigger and bigger, then the Excel is a problem, for sure.
But for a starting point, it's still one of the best tools to use. I mean, let's not let's look at it from the positive side. It's one of the best tools because you can easily model something.
Yeah, but here when you look at these specific examples, so you look at the tumor growth. So you have tumor patients from the past. You monitor over 5-10 years, sometimes shorter because the survival rate is not so high. So you try to make these patients to one average patient, and for sure, first of all, when you want to condense all these people to one person, you need other tools in Excel for sure. But if you come to this stage, then you can use Excel. Yeah.
But I think, the message for me in there is it's not difficult to get started on this journey as an organization. So
definitely.
It doesn't need tremendous amount of investments upfront. So the simplest thing to start is you get a data scientist, use Excel, get started right now, and then when you progress your organization on that journey, you can always invest in more sophisticated solutions. But the first step is pretty simple.
Yeah. So this is actually, I would like to call it, we are living in fantastic times now because everybody can now do fantastic research together with this computer. from home. So because if you need more computational power, then you can use cloud solutions, for example. A lot of companies I see are virtual now, even in the drug development space. So they do all these kind of validation computationally, and then they select drug molecules, buy them, test them in contract research organizations, and then they bring it into the clinic. So this is a lot what you can do computationally. So yeah, as it said, so software is eating the world.
I mean, these are fantastic words at the end of the podcast that you said we're living in the best times ever. When I think back to the 80s, when I had the question, it meant traveling the whole world to get an answer from an expert. Today, we have a Zoom call. I can ask you my questions about artificial intelligence in drug discovery. I can read your posts on LinkedIn where you give away a lot of information for free. And my last two questions to you on this podcast are, is there one topic that you would like to talk about at the end of the podcast that I didn't ask you questions about it?
Yeah, for me, it was just only important to highlight that, in this way we can wrap it up here. So wrap it up. The important part is we have all the hype, there's all this ligand ability, this Alphafold that we predict now, the protein structure, and can do molecular modeling in a better way. But actually the revolution is done now in drugability. So how we can select the right drug targets, how we can find new indication, especially for orphan diseases. This is so tremendous. You can imagine about this. So now you check in your 2 million, you simulate this, and then you get a good model for just 5-10 patients worldwide. And it's then much more easier to convince regulators to conduct a clinical trial with 3 or 4 patients when you have this evidence found in big population data. And of course, this has an impact how we conduct clinical trials. So how we think about causality changing our clinical trials tremendously.
Great points at the end. My final question to you then is, when someone is motivated after listening to the podcast with you to get started on the journey and would like to include your team in their studies, or on the other hand, an investor would like to explore ways to invest in your other ideas that you are about spinning out into separate companies, or do it in your company, what's the best way to reach out to you?
Yeah, so just drop me an email. So I'm always keen to talk to people. So it's always interesting to learn more, because in the end, it's a people's business. So if you have a problem in the area that you're not sure about the efficacy of your compound, you look for a new indication, or you want to check the toxicity, just drop me an email and we will have a look. And if you are in general interested in this topic of how now drug development changed from more reductionistic to holistic science, I can also recommend my book, Chemical Biology, where I described parts how now the whole industry is changing based on these new technologies coming in. Because when you look at CRISPR-Cas, for example, or artificial intelligence, so that was not there 15 or even 10 years ago, and it's completely changing now the industry, and this is extremely exciting now.
That's true. With your consent, I will post your email address, your LinkedIn profile, the link to your book on Amazon. I think it's on Amazon also, so it's easy to.
Yeah, you can book it everywhere. So it's the publishing house is Richard Springer, but also on Amazon. Yeah.
Super. I will post these links in the description to the podcast so people can find you. Marco, thank you very much for this insightful conversation about artificial intelligence in drug discovery. I learned a lot of new things. It was amazing talking to you and listening to your answers. I wish you and your team a happy weekend and good luck for your future and development of your ideas.
Yeah, thank you much. And yeah, thank you for having me here. So it's I always really like it when people ask questions, because then we need to be on the right track, And as I said, so for us, it's really important to bring these new technologies really into the field, because we see a lot of value for people outside. And it's so important to that people know about these chances we have here. And that's why I'm so happy that I could tell a little bit about us and our technology with you today.
Thank you very much, and have a great weekend. See you soon.
Thank you.
Bye-bye.
Too.